Welcome, everybody, to our last Innovators Workbench of the season, which is pretty exciting. I'm Josh McHour. I'm the co-founder and director of Stanford Biodesign. It's a pleasure to have you all here. And this is a very, very special event for us here for a lot of reasons. It's a pleasure to have Dr. Ted Love with us, who chairs the Biotechnology Innovation Organization, which is really sort of the advocacy group on behalf of all biotech. And just to frame out, uh, many of the reasons why Ted is here. About 2.5 years ago, we, on the basis of a lot of feedback from our community, realized that biodesign could really be useful across all technologies. And we really are. We always claim we're technology agnostic, and now we really are. And so, led by Michael Ackerman and a number of folks who contributed to that, we've really recreated our curriculum with a sensitivity and awareness of creating opportunity in biotech, as well as medtech, as well as digital health and diagnostics. So it's part of this broader effort. And so very, sort of, I'd say, emblematic and an exemplar of that effort is having Ted here, who really has an amazing story, as you're going to hear and it told tonight. Ted was already had an impactful career as a cardiologist, then moved to product development at Genentech, and then later Theravance and led several biotech organizations, Nouvelle and Global Blood Therapeutics, to become ultimately a chair of bio. And all of these experiences have given them tremendous insights in this space. What's cool about Ted's story, as you're going to hear, is just the way that he took on every opportunity as it came to him, starting from growing up on a farm in Alabama all the way through to where he is today. It's a truly inspirational story. And, I think also as a part of that, and also synchronous with Biodesign's goals, is really a focus on health equity. And so that I'm sure that Uday will bring that out tonight. So Ted is going to be interviewed tonight by Uday Kumar, who's a senior advisor here with Biodesign on the faculty. He's actually from our 4th cohort of Innovation Fellows. Fifth cohort. Oh, that's right. That's right. And following his fellowship experience, has just had a phenomenal career as an innovator with iRhythm, which is now a very successful publicly traded company. Really recreated the entire space of arrhythmia tracking, and now leading a company called Element Science, which is now kind of recreating this whole area of wearable defibrillators. It's very, very exciting. And of course, Uday is a cardiologist himself, and so Ted is in good company. So it's going to be an amazing story. I won't talk much more except to say thank you to our sponsor, Wilson Sonsini, Goodrich, and Rosati. And a special thanks to Deja Hill and Awal Ado for creating this amazing environment here tonight and setting this all up and getting all of you to connect online as well as in person. And if I can just put a reminder in, please silence your phones, because this is going to be recorded for posterity. And of course, we have an online audience too. And I'll say, take it away, Uday.
All right.
And thank you, Ted. Thank you.
Thanks so much, Josh. As Josh said, I think we're entering a new phase of biodesign with Josh's leadership a couple years ago, thinking about really how do we expand the toolbox of solutions beyond medical devices, digital, AI, and other things. But part of that, and why Ted's here, but there's also successes. There's always this underlying story that oftentimes doesn't get airtime. We just see the success, and I think that's what we're going to explore. And so Ted and I used to work together at Third Rock Ventures for many years, and I've known Ted for a long time and admire his career. And so I think, why don't we start with where you grew up in, Alabama, and then how did you get interested in medicine and science?
So I always like to tell people in retrospect, because I didn't think about it that much, but basically I grew up on a farm, 30 acres, in the Jim Crow South. When I was growing up, George Wallace, who was a very famous bigot, was our governor. Our schools were segregated. Um, but I always tell people I was a very happy kid. Had 7 siblings, so there were 8 of us living in a very small house, which recently just tore down. It's very small. It's about 800 square feet house. But a lot of love, a lot of support. And I was lucky, Uday. I loved learning. I loved school. I really liked things where there was clearly a right or wrong answer, or at least I thought there was a right or wrong answer. So I did okay in things like English, but math, science, things that I thought there was a clear right answer were the things that I gravitated to. And in our community, the one person that I would see that was doing really well and was really respected was our doctor. So pretty quickly in life, I said, well, my background is maybe well suited to do that, so I'll become a doctor. I made that decision probably when I was 8 or 9 years old, and I never deviated from it, which shows you how strange I probably am that I commit to something at that age and just never look the other way.
So maybe building on that, so how did you then think about, leaving the South to go to school? And then, we'll get to kind of cardiology and all in a minute, but those years of education.
So leaving the South wasn't a big issue. In fact, I was probably happy to leave the South in a lot of ways, but not, so much for negative reasons. It's just that I thought I had had a very limited and sheltered existence, and that was accurate. So I was really anxious to get out and see what's going on out there. And Haverford, which is where I went to college outside of Philadelphia, was a big thing for me. I had only been on an airplane once prior to that. Obviously, I'd never been away from home so much. But I always joke also, the other thing I didn't know is the difference between a Lawrenceville and Exeter and Andover college prep from what I had. I didn't know the difference, but I found out really quickly that these kids really were prepared for Haverford, and I really wasn't prepared. So I really had 2 options pretty quickly: either get very busy and catch up, or not become a doctor. And I committed to catching up, and it worked out fine. It worked out well.
Yeah, clearly. So maybe there's a theme that's going to go through our conversation about, as you now encountered, kind of really well-prepared students from all different backgrounds, how did you view yourself in, something we talked about when we've chatted in the past about not being able to succeed, that you had to kind of make it to the next level because of, it's an opportunity and you've been given this?
Yeah, I mean, I can definitely remember, some degree of fear that I could fail. I became extremely close with my chemistry professor. My wife and I have actually started a program at Haverford now that's named after him and me, and we've been given other scholarships to the school as well. But I became very close to him for 2 reasons. One is that chemistry was the subject that was most likely to wipe you out as a freshman at Haverford, and it looked pretty promising to wipe me out. So I took advantage of something he offered on the first day, which is I will have as much office time. I will spend as much time with any of you as needed. So I pretty quickly said, I'm there, man. I'm going to be in your office a lot. In fact, I would not only go to his office, I would sometimes require so much attention that I'd go over to his house and have dinner with his family, and we talk chemistry. So I never really became like his star student, but I think, before Claude died, he said I was one of his most proud students because it was clear that I was so far behind And I was definitely fighting my way into the, upper quadrant of the class. I wasn't number one, but I was able to fight my way from the bottom to the upper quadrant. I didn't love chemistry that much, honestly, but and it didn't love me for sure. But I did get through it and managed to get grades good enough to get into medical school.
Yeah. So then, you were at Yale for med school, correct? And then you went to up in Boston for obviously residency in cardiology at the MGH. So somewhere along the way, even though chemistry wasn't your friend, as you said, you did gravitate back towards basic science. So tell us a little bit about that, because I think sometimes it's not natural, like, well, I wasn't good at that, wasn't my friend, but then you did get back into like sciences, and obviously chemistry's played a role in a lot of the biopharma companies you worked on.
So Haverford, even back when I was in college, a lot of schools actually in the 70s, they were teaching biology, kind of around physiology, stuff like that. Haverford was already doing molecular biology. So when I went to Haverford, I was a molecular biology major. So I was sequencing protein, sequencing DNA. I was doing, very cutting edge molecular biology techniques. So when I went to Yale for medical school, Yale was a medical school, maybe even the only medical school that requires a thesis. So for those of you who are PhDs, believe me, we're not at your level, but we did have to do a thesis. And different people did different theses. The ultrasounds that we do to date the age of babies, that actually was a Yale medical student thesis, where there was this theory that you might be able to track growth and date children in utero using ultrasound. Worked out pretty well. My thesis was not that important. It was not that impactful, but it did get me to apply my molecular biology. I was doing some research in cancer. I published my thesis. It's still in the Yale library if you ever want to go and read it. It's a short read. It's not like your thesis, I'm sure. But that got me, back in the lab, as you said. And then when I went to the Mass General, I did the standard internal medicine training that you do before cardiology. But I had the fortune at the end of my internship to be called into the head of medicine's office. And of course, I immediately assumed something was pretty bad going on. But I sat down and John Pott said, every year we ask about 2 or 3 people to fast track. I didn't even know what it was. I thought it was like fast way how to get out of here. But the fast track meant that you were basically guaranteed any fellowship that you wanted. And I wanted to go into cardiology. And that was probably the most coveted and hard to get position at the Mass General. But in order to do this, you'd have to commit to do some basic research in between. So you do 2 years of internal medicine, you do 3 years of basic research, and then you do your 2 years of clinical training and you're done. So again, that commitment got me back into the lab, and I actually went out and got grants. I won a Robert Wood Johnson. I was reasonably well funded, but I had a lab and I had people working for me. And I'll get to this and maybe end here. I did learn that I could do this and I could publish, but I didn't really enjoy it that much. And one of the things I realized is that I get a lot of energy out of interpersonal interactions, and the lab really wasn't doing that for me.
Tell us the story about the Super Bowl Sunday.
Well, so the most striking example was one day I was in the lab and I was doing some cardiac research using ESLs, actually looking at hypertrophic cardiomyopathy. And I'm in the lab and I'm really quickly feeding my cells, trying to get out because the Super Bowl is that day. And one of my lab mates says, you look like you're in a hurry. Why are you rushing? And I said, but the Super Bowl is going to be on in 2 hours, And she just looked at me like I was completely crazy. And I thought to myself, you really are not in your element. You're not getting the kind of I want to be around people that want to bet on the Super Bowl and drink during the Super Bowl, not just focus on being in the lab. So that wasn't a bit of an extreme, but it was a real wake-up call for me, that getting out where I would use kind of more skills just than my intellectual skills was going to be important.
And if you can't already tell, Ted's a very personable guy, and I think that piece of it, sometimes when you're a place, I mean, this is Stanford, and a lot of obviously very accomplished students and academics here, but you're at man's greatest hospital, you're on faculty, you have grants, but you're thinking to yourself, I'm not fulfilled, and I'm using the IQ but not the EQ. Tell us a little bit about that, because oftentimes students and all feel like, I'm here, I'm doing these things, like I should just keep going. Tell us about that, because that's an important lesson that you oftentimes don't see. And I have a lot of friends at HMS too who probably haven't yet learned that.
Yeah, it was painful because so there was a provoking event, by the way. The provoking event was my mentor at Harvard, Ed Haber, was recruited to become president of R&D for Bristol-Myers Squibb. So I loved him like a father. And he was just such impactful person on my development, my career, my self-confidence, all sorts of things. That, the immediate thing was to do what he wanted, which was to come to Bristol-Myers Squibb. I did kind of feel like I should not automatically just do that. I should revisit, should I get out there on my own now? And so I looked at a lot of companies. I looked at all, I didn't even know what VCs did. I talked to VCs. I didn't know what, equity, and private equity, I didn't know any of this stuff. But I kind of looked at a lot of things, and I finally decided that Genentech was a really cool place, and they wanted me to come very badly. And I just thought it was cool because they were going to use immediately a fair amount of my cardiology expertise. And I was going to gain a lot in terms of this whole new world of how do you go about going from an idea to actually having a molecule that you could file an IND and move through clinical trials and get approved and launch around the world. I didn't know any of that stuff, but I did have the basic scientific underpinning to interact around a lot of the, at least the preclinical and the preclinical science. So Genentech was a great choice for me. I learned a ton at Genentech, and I had a lot of fun, and nobody ever complained about me rushing to get to the Super Bowl.
So just taking a little step there about, you mentioned Ed Haber, and then you mentioned your chemistry professor. We talked a little bit about mentorship and the importance of mentorship early in your career. And I also asked you, and we were chatting about, how when given an opportunity, you felt you had to take it.
Yeah.
In all of these situations, did you ever, we talked about, did you ever look for you were a Black cardiologist, and there weren't that many at HMS at that time. Did you ever look for Black mentors or others who kind of maybe have had some of your experience? Because I know it's paradoxical. You were saying that, well, if I didn't take the opportunities given, that actually might have had a bigger impact on those coming before, coming after you. Can you maybe explain that to the audience a bit?
Yeah. So, just starting with the mentors, I've had tremendous mentors, and I do think mentorship is important to be doing when you're in a position to do it. And I think when you're going through your career, it's wise to get mentors, because the simple way to say it is that these are people that have basically traveled the road that you're on, and they can give you insights just very naturally about what you're going to face, because they faced it 5 years or 10 years ago, and the road is very similar. So I've always had great mentors. I've never had an African-American mentor. And I say that importantly because I just don't think it matters. What you need are mentors that care about you. Now, and I was always able to find amazing people like Ed Haber and like Claude Wintner, happen not to be African-American, but they really cared about me and they cared about my development, and they were extraordinary people. I mean, no doubt about it. in every way. I did, Uday, occasionally have mixed feelings about whether or not I should have gone to, Howard rather than Harvard, or gone to Morehouse rather than Haverford, because those places all kind of wanted me to come. And to some extent, I felt a little bit like I'm kind of ignoring them, if I don't think about going there. But I was also very much saying to myself, I really want to make sure that I'm taking on the highest level of competence. In many ways, I was a bit focused on make it harder on myself. Don't make it easier on yourself. Make it hard. And so I felt like Haverford was going to be a place where it was going to be hard. The kids were competitive, academics were going to be tough. I was also going to be tremendously in the minority. I think there were 5, Haverford was all male when I went there. So there were 5 Black guys in the class, and we were it. And but you know what it actually forced me to do, Uday, is, and I even done this in high school. I went, when I was in Huntsville, I was president of the student body. The majority of the students were not Black at my school, and at Haverford, I eventually became president of the student body. I was the first Black person ever done that, which is not a great statement because there have been so few Black people that come to Haverford. I didn't have much competition up until then, but it forced me to make sure that I dealt with people really, really genuinely, irrespective of superficial things. I was really trying to figure out, is there a relationship here? Do I respect you? Can I help you? Can you help me? It was just, they were relationships that by definition, I was not looking at skin color or around mentor selection or around friend selection or anything. So I ended up, I think, coming out of it being what a lot of people talk about. And that's why in retrospect, I'm kind of happy I didn't go to Morehouse or to, those are great institutions. And maybe now, I'm thinking more about philanthropically supporting those institutions, even though I didn't go there. Because I think there's a role for them, but I think I was a person who, it probably wasn't the best place for me to go.
I mean, the story is an amazing one about you focus on excellence, and you were kind of a pioneer and use the excellence to take you. And, maybe we kind of fast forward a little bit to the next step, like when you went to Genentech. And so Ted's very, very humble about this, but he was really involved in a lot of drugs. I mean, Rituxan, Herceptin, Activa, Avastin. I mean, these are, for anybody who knows those drugs, these are big drugs, all the way to the head of the product development committee. One of the things that when we talked earlier that struck me, that how you got through all those, was that you actually narrowed your focus to the beginning. You said you were given the choice of doing things. You could be the head of clinical and head of research, but you chose research. Tell us a little bit about that, because I think oftentimes here in the Valley, a lot of young people want to go broad, do it all, but you chose to focus at that stage in your career when you were making the transition, and it turned out obviously to be very good for you longer term.
So one of the bits of advice I give people, and I'm not sure if it's good advice, but I give it consistently, is when you take a position, you probably want it to be about 80, 85% something you can really do. 15%, you got to figure it out. That's fine. And in fact, that's kind of stimulating. But a job where it's 15%, you know what you're doing, and 85% you got to figure out, I'm very cautious about those jobs. So when I went to Genentech, as I said, Genentech really wanted me to come, and they knew that I was talking to Merck. They knew I was talking to Syntec. I was talking to a couple of other organizations. So they offered me a dual appointment in basic research and clinical. And it was very exciting to your ego to hear that. But after sleeping overnight, I said, you know what? I think what they're offering me is an opportunity to fail twice. I mean, not on purpose, but I'm going to be competing against, the scientists in the labs at Genentech who are brilliant, and I'm going to be competing against the people in clinical development who are brilliant, and I'm going to be putting in half the effort in both arenas, and I don't think I'm going to win. And to your point about excellence, my view was and it wasn't really just, I always wanted to be the best, and I figured if I was going to be the best, going half-time wasn't going to work. And I don't think it would have worked. So I think that was one of the best decisions I made, was to turn down the dual appointment that on the surface looked very attractive. Art Levinson offered me many promotions early in my career at Genentech, and I turned them down because I kept telling Art, look, I don't want to be in charge of all these people who look at me as a guy who got these promotions so quickly and he doesn't even know what I do. So I wanted to make sure I knew how to do clinical trials. I knew how to manage a CRO. I knew how to negotiate with the FDA. I knew all that stuff, and it took a while for me to know it. And fortunately, that was fairly coincidental with Art becoming CEO. And when Art became CEO, he basically said, I'm not asking you anymore. I'm telling you, you're going to take this position. You're going to run this department. And I, of course, obliged.
Well, I think it's an important lesson, because I think, especially someone who hires a lot of people just like you, we oftentimes have young people who are ready for the next step before they're really not ready for the next step, because there's a lot of, battle scars you need to go through to really become excellent something. And I think Art is someone who recognized that. So maybe why don't we spend, because Art is another example of mentorship and kind of that you had, maybe for the audience, everyone who doesn't know who Art Levinson is, maybe a moment on him, and then how you got close to him, because I think the other thing that you mentioned earlier was about how you treated people with openness and honest opinion.
Yeah.
Because there's a story we chatted about anti-IgE and Xolair, and talk about that, about you established relationships where you could challenge the conventional wisdom and people listened. And maybe start with Art and then that.
So Art became, before he was CEO, he was originally head of research. Well, he started out, obviously, not head of research. He started out as a postgraduate school at UCSF, and like everyone at Genentech, I think in the early days, worked on cloning insulin. Eventually, he became head of research, and then he became head of research and development. So I was in drug development, so the D of R&D. And because when I turned down the dual appointment, I did tell research, I love what you're doing, and I'd love to kind of be involved in research. So we basically came up with an agreement for me to spend, I think it was like one morning a month in research. So I'd go up, and I didn't do anything in the lab. Nobody worked for me. But I'd hear about all the cool stuff going on in research. And, sometimes I would tell them, I think this is stupid. I don't, I think it's clinically useless. But, they wanted to hear it. I mean, I'm not sure if they always wanted to hear the negative news. I think they wanted me to say, this is brilliant. I think this is going to solve a lot of problems for the world. But I was very blunt, and honest, but, sympathetic. So Art also basically said, I want you to meet with me, at the same time. So I'd go down, and Art would ask me a lot of questions about not so much research, but about development and how things are going. And I had the very, very good experience at Genentech, where the things that I worked on worked really well for the company. And a lot of the other stuff was not working well at Genentech. This was back when Genentech was not good at developing drugs. In fact, we were not very good at all. So Art was beginning to kind of try to understand, why is it that the little stuff you work on, and some of the stuff I worked on was really big. Like one of the first trials I did was 40,000 patients, and filed that with the FDA. And we actually got something the FDA almost never gives out, which is a superiority claim. The label literally said, you're superior to your competitive drug. They really do that. And by the way, that was a whole strategy of how I negotiated that from the FDA. I basically decided to make it the FDA's idea, and they fell for it, and they gave us a superiority claim. But Art was mostly focused on that. And then when he became CEO, it really, I think gave him a lot of confidence that that I know this guy and I know what he's going to do, and I trust him. So I did have the job pretty quickly of re-engineering a lot of things that we were doing. Like the first Herceptin trial would not enroll. We enrolled 12 women over 12 months at a cost of about $12 million. I remember all the numbers. And Art called me like a week after I had the job and says, "Why isn't the Herceptin trial enrolling?" And I said, "Art, you gave me this job a week ago, man. I'm working on it, but I'm going to require a little bit more than a week." And as I think you all know, we basically shut that trial down. We re-engineered a new study, 450 women, very striking data, and Herceptin is one of the most transformative drugs ever made, I think. And we were on the brink of killing it because we couldn't generate any evidence. And there was a lot of, I don't know, you guys are too young to remember this, but there was a lot of anti-antibodies back then. People didn't think antibodies were going to be therapeutically relevant. And Genentech was pursuing quite a few antibodies, and a lot of people thought we were crazy. Turns out antibodies have become pretty useful once we solve things like HA and we solve problems that we didn't even comprehend when antibodies were failing for reasons that were solvable long term.
Well, I think, what's remarkable is that the progress and all the drugs that you had, but a thread through all of this is kind of your relationships with these people, to basically take your advice, listen to you, and then because of that, I think we'll get to it at the end, but the EQ side of your skill set clearly was not getting enough exposure at Mass General. So at this point in time, you've been at Genentech, you've been very successful, head of product development, and you're being bombarded by leadership opportunities, but you decided not to become a CEO again, whereas you think by this point you would. Tell us a little bit about joining Roy Vagelos at Theravance, because again, it was one of those examples of 80-20, maybe not yet ready for CEO, and I think this is unusual for this audience, and most people, I just want to go there right away.
Yeah. So I was getting a lot of calls from people in the venture community who were basically just saying, the word is that you're a very good leader, and we think you could, help us build a company, run a company. I was always kind of nervous in those conversations because I was kind of thinking, I don't think I know how to build a company. I didn't build Genentech. We don't raise any money at Genentech. We don't, I don't build the board. I mean, so there were so many things that I thought I had no clue about. So I just continued to turn things down, really with the view, probably honestly, Uday, that I'd never leave Genentech. And then one day I got a call from Roy Vagelos. So Roy had obviously been enormously successful at Merck. I knew Roy because Roy would occasionally come to the Mass General. He trained at the Mass General in cardiology also. So I'd met Roy at the Mass General, and we agreed to meet. And Roy, he's very Roy-like. He basically said, we're going to build an amazing pharmaceutical company on the West Coast. It's needed. And we're handpicking everybody. And we've handpicked you to run drug development. We have a former Merck guy who was running research. So I actually thought that made a lot of sense to me because, number one, I said, well, Roy is there, so I can learn a lot, from Roy. We already have a CEO, and I can learn a lot from that CEO. The company was only like 10 people at the time, so I'll be able to see. Yeah, I'll learn what a startup is like. So that made a lot of sense to me as And I didn't necessarily think of it as an intermediate move, but it did become an intermediate move because the next call I got about 3 years later was George Rathman when he was running this turnaround that was in terrible shape. And he basically said, Ted, you know, at my age, I don't have the stamina to turn this thing around, but by reputation, you could do it and we could be a team. And that was my CEO Academy. It's a terrible company, by the way. I don't recommend turnarounds, but I figured a turnaround would be really hard and I would learn a lot. But the real reason for me doing it was I said, this is the George Rathman CEO Academy. And almost nobody is, you know, how many students can get into that? So, and I've been offered a position. So, and it was worth it. It was worth it. I didn't make any money. I think I lost money actually in Nouvelle because I invested my own money. George invested $50 million, but I paid him back. And I paid all the, I didn't pay all the investors back because our market cap when our drug failed went from like 1.1 billion to like $100 million overnight. That was painful. That was my first big.
That's Novello, obviously.
Yeah, that was Novello. That was my first big punch in the gut.
But this is already kind of mid-career, and I mean, I think again, part of the surprising part of the whole story was you realized like you had to go to industry to learn industry, so you went to a big company. Then you went to a startup to learn what a startup is. It's very not conventional. Most people are like, I just go to a startup and I know how to do everything. Versus, you know, you're 80-20. You're learning, and then you not only took a CEO role, but you took one at a turnaround, so you could learn about like what are like when it's challenging. So I think it's a unique thing that we typically don't hear, but it's really important for a lot of folks who look at your success that it didn't just happen overnight. So Novello, you told me that was like a labor of love and it took a ton out of you. So after that, you went to retirement for a little bit. So tell us, you know, what happened during the retirement that you finally got called back, you know, to go to Onyx, just briefly before we get to GBT, because I want to really talk about GBT for a bit.
Yeah, so Onyx is pretty simple. I was looking for a job. I was just sitting on boards and doing things. And Tony Coles, who I trained with the Mass General, became CEO of Onyx. And Tony and I had been friends from the Mass General. Our wives had been friends. So Tony and his wife were at our house for Thanksgiving dinner. And Tony begins to tell me the story that all the investors are saying, you bought proteolytics, you got this drug. It's now called Kyprolis. It's now in the market. It was called carfilzomib back then. You're never going to get it. You're never going to get it approved. The FDA is not going to approve this drug, but you have a buddy that's magical at this stuff. Why don't you get your buddy to come out of retirement because he'll get it approved? And
but he wasn't asking to be CEO.
No, he wasn't asking to be CEO. He actually said to me, "It's a little embarrassing to ask you to do this because you've actually have more experience of being a CEO than I've got. But I've got the CEO job. I need somebody to come in and run R&D." And I basically just said, look, I'm not looking for a job. I don't want a job. But in some ways, honestly, Uday, it was kind of, it struck the right chord because I really had so much pain in failing as a CEO in that job. I wouldn't say I failed, but the initiative failed. And it was so painful. I told you the story. It really felt like, you know, you're the NICU doctor that nurses this baby that nobody thinks can survive as a preemie through months and months of care, and then you're discharging from the hospital and you get a call that the baby arrested in the lobby of the hospital and died. It literally was that bad. So I wasn't looking for a CEO job again. I wasn't looking for any job. So in some ways, the fact that I wasn't the CEO was kind of gratifying. And I figured I do know how to get drugs approved. And that's really the big need here. It took me about 2 years, but we got it approved on an accelerated basis, which everybody bet we couldn't do. And ultimately Amgen bought the company for, I don't know, $11.5 billion or something shortly afterward. But after the drug got approved, I retired. I just said, I came here to do a job. I've done my job. I didn't want a job. I don't need a job. I'm gone again. So I went off to retirement again.
So I think, again, another unique aspect of the story is, most CEOs, once they're CEO, can't imagine not being CEO again. But I think, what's thematic through all of this is you've really been focused on the work and the people, not the title. And that's really, really an important lesson for everyone out there, because it seems like, obviously you've been able to get fulfilled along the way. More importantly, you've also been able to take some of those struggles from like a Novello and then translate it. So now you're in your second retirement, just hanging out.
It's going really well.
And so let's, well, this is the third retirement technically. Yeah. So then let's talk about a little bit about Global Blood Therapeutics, which is a really, really important company. So for those of you who don't know, it's a company that was started at Third Rock Ventures by someone we know very well, Charles Holmesy, who found some research that that suggested that there's a opportunity here to take a disease that disproportionately affects Black individuals, African Americans, and really make a difference. And so you were in your second retirement, obviously did very well from Onyx, among other things before, and the sale to Amgen. But you said something to me that was really interesting when we were talking before, that when you heard about this and then you thought about the science, we'll talk about the science in a minute, this was the thing you'd come out of that second retirement for, not only because, it sounded like, but you must do this because of the nature of the patient. Let's talk a little bit about GBT and sickling a little bit for the audience, and then talk about why this was the company that made you come out of retirement.
So I initially told Third Rock I would not do it because I had promised my family that after my career, where there was so much neglect for them at the, because I directed so much time to my career, that I was going to focus on them and only them now. So I told Third Rock I couldn't do it, but I would agree to take a board position because I thought the science was compelling, and I care about sickle cell disease, and I'll help them. So over the course of a year of being on the board, of course, the CEO search was unsuccessful, probably because they didn't want it to be successful. And the company was generating more and more data that really said, this is going to work. So I'll step back a little bit. I don't want to go through in too much of a lecture, but for those of you who don't know sickle cell disease, it's a point mutation. It's a single amino acid change on hemoglobin. And hemoglobin is an allosteric molecule, which means it slightly changes its shape. And there is one of the shapes actually essentially hides the mutation. So the hemoglobin behaves pretty normally if it's in the so-called R state. So the idea was to make drugs that would bind the hemoglobin and make the hemoglobin prefer to be in the R state, in which state it would be subtracted from the polymerization that drives sickle cell disease. So this is a pretty cool idea. And there was actually other data with really terrible kind of non-drugs like sodium cyanate, for example, that suggested this probably could work if you made a really good drug. So the concept was really, really cool. What happened as the company began to generate more data and began to take it into sickle mice, you literally in days see these sickle mice transforming on these therapies. And I called my wife as I was flying to Seattle for a board meeting. I'll never forget, I was in a taxi. Nobody uses taxis anymore, but this was back when, before Uber, before Lyft. I was in a taxi riding in Seattle. I called my wife and I said, honey, I am beginning to wonder if I really don't have to do GBT. I think this is going to be transformative for sickle cell disease. I know how hard drug development is. I know how difficult it is. I can do it, and maybe I should do it. And my wife said, Ted, we're not going to talk about this. Go to your board meeting. Come on, we'll talk about it. That was not a good start, I thought. So I get back home, and I walk into the house, and we sit down for dinner, and my daughters began to talk, and they said, Dad, we've already talked about this. We know who you are, and we know about sickle cell. And we've decided that you must do this. So it went from you can't do it to we feel you will be a very unhappy person if you don't try this, and we don't want you to be an unhappy person. It's a great family interaction. So they not only gave me permission to go back and work 80 hours a week, but they a lot of wind at my back. So I went back and I told Third Rock, if you guys still want me to do it, I have permission to do it, and the rest is history.
So what was that little bit? I mean, you mentioned that your family was the one who was pushing you. You must do it. How did you feel about must do it given this particular disease state? Because I know we talked about that.
Oh yeah, I was there. I was there, but I was weighing this kind of almost moral imperative to do something for sickle cell patients versus the moral imperative of my family has been behind me through all the Genentech, all the travel to Switzerland, all the, being a CEO, particularly for turnaround, it's brutal, right? You know this stuff. So getting their permission was the only way I was going to do it, even though honestly, they were right that if they didn't give me permission, I was probably going to always feel like I really should have done this. I'm sure my marriage would have been fine and we would have continued to be happy, but I think everybody feels good that the right decision was made.
Yeah, so as you kind of, ultimately getting to the sale to Pfizer and getting the drug out there, tell us a little bit about the patients that you met along the way. Because I think, as I'm a minority leader treating a disease of individuals who kind of look like you, I know you shared some stories about that.
Well, I mean, the delivery of care to sickle cell patients is fraught with racism. I mean, just beyond imagination. And I, even though I grew up in a segregated Black community in the South, I didn't know what sickle cell disease was. Nobody talked about it. It wasn't brought up in my education. I really didn't know anything about sickle cell disease until I got to medical school. And then when I go into the hospital, I start to see how these patients are treated. And my big contrast that I tell people is that if you showed up at Yale with cystic fibrosis, we had a lot of sympathy for you because we knew you were probably going to die in your mid-20s. You were going to have all these terrible infections. So we were very accommodating you. We had a special part of the hospital that we admitted you to. You always saw the same people. You got your chest physiotherapy back then. We weren't changing the outcome that much, but we were treating, I thought, these patients with the right kind of treatment that they deserve, even though we weren't changing the outcome. Now, next patient come in with sickle cell. Ironically, very similar profile. They're going to die in their 20s. They come in with this excruciating pain. Yet, the system essentially said, we don't know if you're really having pain. We think you may be drug seeking. So there was a lot of manipulating going on. Some people would even give sickle cell patients saline and tell them it was morphine to see if they would say, oh, my pain is better. And that would be a test for them to say, oh, yeah, this was a malinger. So that kind of stuff was very common in sickle cell. And sickle cell patients knew it. As I got to work with sickle cell patients more, of course, I heard that over and over again. I heard from sickle cell patients that before I go to the emergency room, I put on my best clothes, I put on makeup, because I know it will mitigate the risk that I'm treated as somebody that doesn't deserve the kind of respect and empathy that was routine for other people. So this bothered me. And while I didn't execute that way with sickle cell patients when I was training, I knew that was what they were dealing with. So GBT was in part an opportunity for me to say, you know what, I can't go and change all these people, but if I could just so dramatically change the course of this disease, then these people won't be facing this stuff. And then as I got to know patients, one of my sickle cell colleagues who has sickle cell would say to me, Ted, going to the hospital is not it's not an act of need. Going to the hospital is so unpleasant for us. It's an act of desperation. And that was part of the reasons that I didn't like VOC as an endpoint for a disease-modifying therapy, because I think it's a healthcare utilization endpoint in a population that doesn't want to utilize the healthcare system for very good reasons. So that's another reason that GBT was so focused on using endpoints that weren't focused primarily on healthcare utilization. We were focused on using endpoints that really did show we were modifying the underlying pathophysiology of the disease, with the idea that that would modify the long-term outcomes, including survival. And I think it will, but we haven't proven all of that yet. But I would tell you this also, now maybe this will point in it. One of my frustrations that continues is that Vertex comes out with a drug that is a rectifier, and it shows that they can make the cilia in CF patients move, and they can mobilize more sputum, and they get it approved on that basis, and everybody gets it. Everybody gets it on the belief that if you correct their cilia, you're going to have probably you're going to have good long-term outcomes. I think that's right, by the way. I think that's right. And I think it's being proven true now. We have a drug that shows the cells stop sickling. The hemoglobins in some cases become completely normal. Patients will tell you, I feel much better. Yet doctors are often very reluctant to prescribe it. There is not the empathy for these patients that exists for many other patients. So GBT was also focused on that. And I'm trying to keep Pfizer very focused on that, because sadly, we need more than the therapy to solve this problem. We need the behavior, the attitude, and that's not easy to change overnight. CF didn't have that built-in barrier to how they're cared for, but sickle cell does, and it's still there.
Well, that's, I think fascinating to think about the inequity in drug utilization, and obviously what you're talking about in terms of patient experience is well understood, but how laudable to try to, well, I'm not going to change the system, but I want to change the pathway for the patients. We're almost out of time. I think we have a few minutes, so maybe just to summarize, and then like to get your closing thoughts. So I think for the audience, particularly a lot of the younger folks in the audience, hopefully you found this story fascinating about all the things we've touched, Ted. You and I have known each other for some time, but if I just had to highlight a few things that I take away when we ever talk, and so one, careers often progress incrementally, and there's struggle along the way. Two, finding supportive mentors and knowing your own limitations at different times is really, really important. And corollary to that is, if you do focus on the work, not the title, that's really, really critical. Spending time developing your EQ side, I think that's probably helped you more throughout all this in terms of mentorship and the relationships that have all allowed you to network to get somewhere. So don't ignore these, getting to know other people, because clearly that's had a huge impact in terms of the doors open. And I think fourth, that you eloquently just talked about, what it means to be a minority leader who's seen treatment of certain patients in a certain way, and that, not just in that position, but those who come after you are watching how you do. Because I know you gave me some examples where, like, if you slipped up, people will be like, yep, yep, we thought he would slip up, but so you were always there. And then finally, the personal enrichment. It's clear in your voice when you're talking about this experience. It was very meaningful to you. So, any other thoughts from you before we wrap up and ask for questions? I mean, it's really fascinating to hear you tell it.
Yeah, I mean, I always like to tell young people that are at the beginning of their career, anytime you're given an opportunity to do something, do it with outstanding excellence. Do it with grace. Do it in a way that that's exceptional, both technically as well as
And
kind
humility.
of personally.
Your humility.
Exactly. And then more opportunities are going to come your way. But you don't have to get out over your skis in your career. You got to make sure you deliver on what. And the other thing I've been telling people a lot is, enjoy the journey. I've been to so many destinations, and when I get to the destination, it's never as important as I thought the destination was. And what I look back is say, what I'm really cherishing is the journey. So enjoy the journey.
Thanks so much, Ted. With that, please help me give a round of applause for Ted. So I think we have a few minutes for questions. Yeah, 5 minutes for questions. And so there's mics here. Oh, right here.
I'm the first one. Okay. Well, thank you so much for the sharing. It's very, very inspiring. Yeah, actually, I have a question about your retirement. So, what did you typically do during your retirement? And did you find any difficulties when you're trying to get back to the workplace? Thank you.
So when I left Onyx, I joined a couple of country clubs, golf clubs, and I said I'm really gonna get my golf game to be impressive. So and it was not good when I started, but it became fairly good. I got my index down to about 10, and I enjoyed it. I play golf almost every day, but I still worked. I was still on boards. Someone like me probably is always going to work at least 40 hours a week, but I normally work like 80 hours a week. So having 40 hours a week to golf, to run, to do things that are with my family is a nice situation. My problem right now is that I say I'm going to work 40, and then next thing you know, I'm working 60, and I'm not playing golf as much. But I'm pretty committed. I'm still playing golf. I also think mentoring is important. I'm going to climb Kilimanjaro in September. I'm doing it to raise $1 million for sickle cell care in Africa, in areas where a lot of the kids die before their 5th birthday, simply because they don't get diagnosed. So this $1 million will go toward early diagnosis and really simple treatment that will help them get beyond their 5th birthday. And then my wife and I are going to give $1 million for care in the U.S. because I think it's good to. We're going to give a lot more money than that, but I thought matching $1 million made sense for that, rather than doing something else. So I'm still focusing on trying to make the world better, because I think that's what our industry is all about. We're all about using innovation to make the world better. I'm at a point in my life where I can do things that other people can't do, and I'm doing it.
One more. Another question? Anyone? Right up here.
Yeah, thank you. Thank you. I reiterate that was very inspiring. So thank you for sharing. So you've had a very sort of broad career. What is exciting you about sort of the next 10 years in this space?
It's a great question. I get asked that question a lot. I just think that the pace of innovation is only going to increase unless we screw it up. I mean, I think things like RA could screw it up. I think some of the things, and I think almost all of these things, by the way, I think people have good intentions. But as we all know, things have unintended consequences many times. So I think if we allow the natural course of things to happen, innovation is just going to continue to rev forward. And we're going to be producing more and better therapies. I think we're going to be inventing more highly effective therapies, more curative therapies. I laugh with Rob Caleb sometimes, and we talk about it because, the FDA is on the other side trying to regulate all of this, and he knows that it's a tsunami of innovation coming. I mean, we're going to have therapies that are genetically targeted toward one person. How do you prove that? You're proving it for one person. So I think it's extraordinary. I think we also have a lot of work to do around climate change. We have a lot of work to do around dealing with food security. And I think science is a way to make all that happen, and it will.
A lot to do in 10 years.
Yeah.
How amazing was that? How about a round of applause for Dr. Love? Thank you. That was truly amazing, and I'm inspired as well. And thank you, Uday. Great job bringing out the story. I want to invite everybody to stick around. We're going to have some refreshments in the back and an opportunity for some networking and more conversations. And thank you all for being here tonight. And thank you to our amazing biodesign team who've made this all happen in this space. It's great to see a standing room audience here and so many people online. So thank you so much. Have a great evening. Thank you.